And the next group is a big group. In fact, it outnumbers, in terms of species, it outnumbers all other organisms on Earth, including from bacteria, fungi, everyone. There are more species of arthropods known than all the rest of life put together. Okay, it's a very, very big group. And so this includes things such as spiders, ticks, mites, and the biggest group of all is insects. Now, in a Linnaean classification, this is classified as a phylum, at phylum level, just to mention it. But as I said, I'm not paying too much attention to Linnaean classifications. So it's a very, very large group. Has a lot of the derived characters, namely, they are segmented, and the segmentation has evolved in animals 3 times independently. 3 times independently. We saw it once with the annelid worms. It actually also with vertebrates, okay, like, such as ourselves. That's the second occasion, but here it's also. So I'm going to go ahead and put in this segmentation as a derived character. And furthermore, they have an articulated exoskeleton of chitin. Now remember, these are ecdysozoans, and so ecdysozoa, what is one of the derived characters of it, is that they have a cuticle, okay, outside of the skin, outside of the epidermis. But in nematodes, that cuticle is primarily collagen. Here, that cuticle, that exoskeleton is still the cuticle. Same thing, cuticle, exoskeleton, but it also, besides having protein, such as collagen in it, it has a structural polysaccharide, namely chitin, okay? And some of the proteins are cross-linked, and so it creates a hard sort of shell on the outside. That's exoskeleton. Serves both as a skeleton for support. However, it's not just only on the outside, it also has processes that go from the outside inward. Okay, if you take open a cockroach, you'll still see these spines and ridges that are on the inside of the body that originate from the exoskeleton, and those are also for muscle attachment. So muscle attachment. And when I say articulated, that means that there's joints, okay? So there's areas of flexibility between that exoskeleton, and you can also see that on their legs. Their legs are jointed. This exoskeleton is protective. They have glands in the epidermis that secrete a waxy substance that gets on the exoskeleton, and that waxy substance cuts down on water loss. And as a consequence, we find arthropods, one of the groups that have, animal groups, that has fully adapted to terrestrial, or very well adapted to terrestrial conditions. We've come across other terrestrial animals, and as we've been going here, for example, annelids, some of them, leeches, and oligochaetes, earthworms, they happen to be terrestrial, okay? But you find them in damp areas. So they're still have to be in the moist area. And also gastropods, they have invaded the land. Snails, okay, the mollusks, they have invaded the land, but they too are not fully adapted to dry conditions. You still find snails in moist conditions, but you find these critters in deserts and areas where there's a great lack of water. So they're more adapted to terrestrial conditions. And that's because of the exoskeleton and also that waxy layer that's on cuticle. Prevents water loss. Now, of course, this problem with growth, that's true not just of insects or arthropods. That's true of nematodes as well. All right? So our apomorphies up here is exoskeleton of chitin. Where else do we find chitin as a structural component? Cell walls of fungi. Okay, cell walls of fungi. So just like the nematodes, the arthropods, they have to molt. But because, and molting is shedding the cuticle and growing a new one. Okay, so they go through these molts. However, because of the exoskeleton itself and all these other ridges and spines from the exoskeleton that go inward, molting in arthropods is a little bit more complicated than what it is in nematodes. They also have legs, or well, let's put it this way, it's not just only legs, jointed appendages, okay? So an appendage is anything that's sticking out from the main axis of the body, and these have joints, they're movable, flexible. Most of the time these appendages are legs, but not always. They have other types of structures, such as antennae, mouth parts, that probably the mouth parts are derived from legs that were around the mouth, close to the mouth, but now they have a different function. And so, jointed appendages. And just showing some appendage here. This here is of a spider. So there's a joint called a coxa, and we don't have to mess with this right now. But also there's other things that are sort of leg-like, but they're not legs, such as chelicerae and pedipalps, which I'll talk about here shortly. Those are also jointed appendages. Now within the arthropods, it's believed that the ancestral arthropod had a pair of appendages for every segment. Okay, and then along the length of the body, some of these jointed appendages have functioned as legs, others as mouth parts, others as antennae, and so forth. But pair of jointed appendages per segment. And this group has a very high level of cephalization, means that you clearly can find a head, clearly can see a head on it. When you look at a nematode, sometimes it's a little bit hard to figure out what the head is. Or an earthworm, it's pretty hard sometimes to figure out what the head is. But on these critters, it's very easy to tell what the head is. Have eyes and other appendages on their head. Some other types of characteristics here, biologically important, is that they have an open circulatory system. And so they have a musculated blood vessel that is not compact. It's a tube on the dorsal side going down the midline, and it's musculated and it beats, and it just takes hemolymph, and it's just keeping it circulated around the body. Other than that, they don't have any other types of blood vessels. Yes?
Is high degree of cephalization anapomorphic?
No, because it's happened so many times that convergently that I just don't count it. It's not unique enough. For gas exchange, it depends on whether it's a terrestrial, aquatic type of arthropod, or if they're on the land or in the water. They breed by means of gills if they are in water. Like most of the things that we call crustaceans, many of them are marine or freshwater. If it's on land, there's various types of respiratory systems. They have, for example, arachnids are spiders, okay, include spiders. Something called book lungs, right? Now this word lung, just like this word gill, we use it as a term for analogy. Okay, they have similar functions, but they don't have the same evolutionary origin. So, unless it's within a taxonomic group, okay? So, book lungs are not homologous to our lungs. Our lungs are completely different, structurally, etc. Insects have a rather unique respiratory system called trachea, okay? And I'll point out later what the trachea happen to look like. Now, right here. Okay, instead of having a particular organ that's localized and that engages with the circulatory system for distributing oxygen and collecting carbon dioxide to get rid of it out of the body, they have this system of pipes that are called trachea that penetrate all the way through the body. And so there's openings to the outside that are called spiracles. So here's a spiracle. For example, this is an insect. Usually they have a spiracle up here close to the head, another one about right there on each side. And usually every abdominal segment in insects, they have an opening to this piping system. So it's like a tube, so oxygen can come in, and then the tube system will branching into smaller and smaller units, and it delivers oxygen directly to the cells in the body. Okay, so and so in the case of insects, their circulatory system plays no role in gas exchange or in the movement of oxygen throughout the body. That's simply done by the trachea. And this trachea is chitinized, so it's of ectodermal origin, and so it has chitin, so that when these critters have to shed their exoskeleton, they also have to pull out all of this trachea as well and replace it. And so it gets to be, molting in insects is quite an ordeal. Okay, so for the classification, I'm going to put up here a group that is no longer around. It's near the base. There's some of the earliest arthropods. Trilobites. They were plentiful, species rich during the Paleozoic. They all died out at the end of the Paleozoic with that Permian crisis. And so some of these got to be pretty big. Some of them were real small, maybe about that big, less than a centimeter long, but others were really quite large. Okay, so they're very species rich. All of them marine. They lived in the ocean. And I'm putting up here, in fact, this is their body is divided longitudinally from head to tail by 3 areas. Okay, so 3 sections, and this is where they get their name, Trilobite of Morpha, right? And that's the only thing I'm going to put up there as an apomorph. Other than that, I just want you to know that they existed, and we're not going to pay any more attention to them. We just pay attention to what is alive today. And so I'm going to keep this as a tritomy. Okay. So I have 3 branches there coming to this one point, 3 different clades. So Chelicerata, these spiders, ticks, mites, scorpions. Wrong way. They don't have any antennae. I'm not putting that as a apomorphy because trilobites also did not have antennae. Antennae are special types of organs that will be found in the Mandibulata. These are derived from appendages, okay, real close to in fact, appendages are real close to the mouth, and they're for chemosensory, okay, taste and smell, all right, that's what they're used for. They're not used to pick up EMTV, all right. Now, the types of mouth parts. Mouth parts in the arthropods are derived from jointed appendages, okay? And so these in the Chelicerata, there's 2 different types, 2 pairs here. I'm counting them as just one apomorphy, namely Chelicerate and Catapults. Okay, Chelicerate and Catapults. And this is looking at a spider. If you're looking at it in front, The pedipalps sort of look like their legs. However, they're not used for walking. They are used a lot of times for manipulating a food item. And these so-called poison things that you would see on a scorpion or on a spider, those are the chelicerae, okay? They, although they don't look like they're derived from legs, but they definitely are, from leg-like appendages. But they're used, usually they have poison glands, and therefore they will inject poison into their prey. Yes?
The tail of the scorpion is not involved with that, right?
What's that?
The tail of the scorpion is not involved at all with that?
Well, they do use it for stinging.
But it's not like a chelicerae, right?
No, no. And all spiders, they do have poison glands. But usually their bites are not serious. It's just like a bee sting. There's a few spiders, like in the United States, black widow and brown recluse, that some people have, there's a few people die from, okay? But most people who get even bit by these spiders still survive, yes.
So I was just gonna ask you about the glycerin. You said that there's spiders whose poison is built to not affect the body, right?
That they what?
Like black widows in the body.
Yeah, they can sometimes be fatal to humans.
Okay, because I was gonna.
But all spiders are poisonous.
Okay.
They all have poison glands. Yes.
Is it a reason because of the potency of the poison, or is it something else?
No, the poison is for subduing their prey. Okay, and they don't chew with these chelicerae. What they do have saliva glands, and they will inject saliva because a lot of times they're eating other insects or other arthropods. That's normally what they're eating, and so they'll inject saliva into the body of the insect that they've caught. And their saliva contains digestive enzymes, and it turns the inside muscles and organs into a mush, a liquid. Then they suck that liquid out, okay? And that's how they feed themselves. There's some insects that do that too. Okay, one group, these are horseshoe crabs. They're chelicerates. There's a sura. So that's coming off of the Chelicerata. These are horseshoe crabs. Their body form of these things have not changed much since the early Paleozoic. Okay, they look pretty much like that. But that doesn't mean that the modern horseshoe crabs are genetically unchanged from the Paleozoic forms. Genes are always changing. They're always changing. So their genetics is probably quite different, and we don't have access to it. But nonetheless, the body form has stayed pretty much the same. The biggest group of chelicerates are the arachnida, okay? And this includes spiders, ticks, mites, scorpions, where spiders are one of the biggest group here. And I'm just putting up there for an apomorphy is that they have 4 pairs of legs. So on the abdomen, there are other appendages on the abdomen, but they are not legs. Now, these segments in arthropods are often fused together to form what are called tagma. Okay, so tagma. This is parts, because these are segmented animals. And in some cases, the segments in a certain region have sort of fused together. We can still find the segmentation on the inside, okay? But on the outside, sometimes hard to see the segmentation. And when we have these tagma regions, they usually have a particular function. Now, in the arachnids, we have 2 tagma. One is a cephalothorax. And another region is abdomen. So the cephalothorax contains the eyes, okay, and mouth parts, other sensory organs, technically the head, and any walking appendages, okay? So all the legs, all 4 pairs of legs, these are 4 segments at the posterior end of the cephalothorax, all right? And Their eyes are simple eyes. And what I mean by simple eyes, a lot of times they'll have 8, 6 or 8. These have a lens, but it doesn't focus. Okay, there's no focusing on it. There are light sensing neurons just underneath that, and these will react to light that's coming in, but the eyes do not focus. And so they really don't see images. They mostly are detecting movement, light and dark. Okay, so shades of light, movement, they can pick it up, but they really don't form an image. Okay, the eyes don't focus. Types of respiratory structures are in the abdomen, on the underside. This is for gas exchange with oxygen in air. They're called book lungs. And the main part of the abdomen, most of the visceral functions happen to be in the abdomen. So that most of the digestive system is in the abdomen, reproductive system is in the abdomen, and so forth. Excretory system in the abdomen. Yes?
Do horseshoe crabs have eyes?
Horseshoe crabs, I think they do, yes.
Are they also simple eyes?
In most cases, yes. But I think they also have something called compound eyes. I will talk about compound eyes later. Okay, so simple eyes, yes.
Are eyes part of the cephalothorax?
What's that?
Are their eyes part of the cephalothorax?
Yeah, they're on the cephalothorax. So this whole region up here, from here to here, that all is cephalothorax. Okay, and on the spider, wolf spider, where the legs are and where the eyes are, chelicerae, all that is cephalothorax. Okay, and these things right here, those are not legs. Okay, these are the pedipalps. So the pedipalps look like legs, but they have totally different function. They don't walk on the pedipalps. And of course that's the Sopra, those are horseshoe crabs.
Here's something.
And what's that?
What's the amber part?
Stingray?
Well, they have algae growing on them.
Oh, I mean, what's the thing in the middle there?
That's a horseshoe crab.
Oh.
Probably a female, and that's probably males.
Mm.
Okay. Very likely. Okay, and just showing little pictures. I'm not going to go over the anatomy, but nonetheless, this here is the cephalothorax. The abdomen is mostly with the visceral functions, okay, inside of it. Book lungs still right there, okay, on the underside of the abdomen. They do produce silk, okay, which is a type of a very strong protein. This is actually pretty widespread in arthropods. So it isn't only spiders that produce silk, but various groups of insects also produce silk for cocoons and other functions. But most of the silk and the arachnids and the spiders is for capturing their food. All right, so the other clade, that's enough for the Chelicerata. The other big clade is called Mandibulata. And here the mouth parts, instead of being chelicerae sort of work in a, well, I'll leave that out because that's not always the case. But nonetheless, this group has mandibles. Their mouth parts are right by the mouth. Mandibles, like right here. Okay, in this ant, those are the mandibles. And they're different than chelicerae. In fact, their evolutionary origin is from a totally different segment that is in the head. Furthermore, there's other mouth parts. I'm putting mandibles here. There's a major one, but they also, behind the mandibles, have another pair of mouth parts called maxillae. In fact, they have unpaired labrum, closes the oral cavity in front, but we believe that it comes from fused appendages. That's just anterior to the mouth, sort of plate-like. And right behind the labrum is a pair mandibles. They're paired, okay, one on each side. And these are moved by very strong muscles that have their attachments way up inside of the head. And right behind them is another pair of jaws called maxillae. And maxillae, they're still used to manipulate the food, hold it while the mandibles can crush it in many cases. These are very leg-like, okay, but they're not legs. They have a lot of semblance, more of a semblance to walking legs. And then behind that, a pair of appendages that are fused together, called labium, okay, and it has, that closes the posterior end of the oral cavity, okay? And the maxillae and the labium, they look very similar to each other, except for the labium, the two halves are fused together, and it closes the oral cavity. When you do the cockroach dissection, when you come back from spring break, I will have you behead your cockroach and remove its mouth parts. Okay, you get to take out all those mouth parts and identify it. Okay, another appendage that is not found in other arthropods, that is in the Mandibulata, is antennae. Okay, and the antennae are once again derived from paired appendages, and the whole function of the antennae is to usually chemical. Okay, they pick up molecules in the air. They have sensory receptors all along the length of the antennae, and so they smell with them. Okay, so this group Mandibulata includes centipedes, millipedes, things that are called crustaceans. That used to be thought to be a monophyletic group, but crustaceans are not. It's paraphyletic because the insects belong there too, yes.
So, before you said antennae, is it chemosensory?
The antennae are.
Yeah.
Yeah.
Is that
Chemosensory. Chemosensory is, well, our organs of chemosensation are both smell and taste. Okay, but they don't really taste with their antennae. It's mostly airborne chemicals that the presence of those chemicals are sensed. But taste and smell are very, very similar to each other. Okay, so one clade within the Mandibulata is Myriapoda. So we have here. And this includes two different groups, centipedes and millipedes. Now, as I said earlier about the mandibles have to brace the muscles that operate the mandibles. There has to be a lot of reinforcement in the head capsule. And there's something called a tenebrium that's there. You don't need to know that word. But that tenebrium is where a lot of the jaw muscles happen to be attached. It's internal into the head. And it takes a certain form all throughout the Mandibulata, except for in this group, that tenebrium has a very special, unique structure. And I'm not going to explain what that uniqueness is. I just want you to know that brace reinforcement of the head capsule for the jaw muscles takes a particular form in this particular group. Okay, different from all the rest of the mandibulates. So that's the major apomorphy for Myriapoda. And another one here. Now this ultrabithorax gene, there are various genes in animals that are called Hox genes. Now these are usually expressed in the very, very early embryo, and they're important for setting up great chemical gradients, usually protein that will diffuse throughout the body. So where the protein is, first of all, if it's produced in the head, then it's going to be most concentrated in the cells around the head, and there will be a gradient going toward the posterior end. The further away you get from where that gene is being expressed, the less concentrated it is. And by means of these gradients in the early embryo, That establishes what is going to be the head of the animal, what's going to be posterior, what's going to be ventral, what's going to be left and right. And so these are very important genes. And they're called Hox genes. There's many of them, many different ones. One of them is called ultrabithorax, okay? And in this particular group, centipedes and millipedes, it happens to have a very, we're not going to worry about antisense expression, but remember that the gene has, the DNA has two strands, sense strand and the antisense strand. We call it template and non-template strand. Okay, so the sense strand is the same as the non-template strand. Okay, the antisense strand would be the same as the minus strand. And so this here is being expressed at a very, very high rate in this particular group of animals. Okay, two different clades here, Diplopida and Chelopida. And Diplopida. The Diplopida, they are the millipedes. All of them are herbaceous. There's none of them that happen to be, predatory. So they're eating usually plants or decayed material, okay, organic material. They appear to have two pairs of legs for every segment, okay? Now I said that paired appendages in arthropods primitively, plesiomorphically, was a pair per segment. So how did they end up with two pairs of legs per segment? It's because in their body, every other segment are fused together. And so these are pseudo-segments. Pseudo-segments that really are comprised of two fused segments. And so they appear to have two pairs of legs. Now this word millipede means millions of legs. They don't have that many legs, okay, but they have a lot. In the myriapods, their tagma is head, and trunk. Okay, so they have a capsulated head at the very beginning, and this is true for both centipedes and millipedes. And then the rest of the body is the trunk. All right? And Chelopida, these are all predatory. So I say here that their body consists of a head and trunk. This is also true of diplopods. Okay, that's true of all myriapods. The tagma happens to be head and trunk. Now, the first pair of abdominal, this trunk is also called an abdomen, incidentally. Trunk, abdomen, same thing. But the first pair of legs on the trunk, in the case of centipedes, are modified into a pair of poison glands. They're still leg-like, but they're stiff, they're not two-jointed, but they're a pair of fangs, and this is what they stab their prey with. So all centipedes are poisonous. They're not too dangerous to humans, except for in the tropics, there's a genus called Zentroides. You don't need to know that, but some of these centipedes can be that long, and some of their bites can be fatal to a human. Some of these centipedes, like right here, it's eating a frog, all right? So it's killed a frog. Most of the time, they're killing other and feeding on other small organisms like snails and earthworms. And they have a pair of legs, one pair of legs per segment. And this is an apomorphy for Chelopida. First pair of legs right behind the head happen to be modified as poison fangs. And when you look at these in lab, after you come back, I'll still have them out, and you can take them out of their jar, put them under a microscope so you can get a real good look at those first pair of legs. And they're all predatory. All centipedes are predatory. Yes?
So one pair of legs per segment is a plesiomorphic.
No, that's a plesiomorph, yes. Plesiomorph. So fused segments in the Diplopoda. That's an apomorph. Yes?
Wait, is it the fused segments that's apomorphy? Or
That
just
is,
like-
yeah, fused segments, but that's why I put up here 2 pairs of legs per segment, because that's easier to see. That's easier to see. But the reason they have 2 pairs of legs per segment is because those are fused segments.
Those are pseudo segments?
What's that?
Those are called pseudo segments?
Pseudo segments, yeah. Okay?
If we were ever given a collision test, and like, we put pseudo-segmented as the apomorphy, or do you want us to specifically put 2 pairs of legs as the segment?
We talked about this
Let's talk
or
about
this.
That one.
Yeah, so 2 pairs of legs per segment is the apomorph.
Okay.
Yeah. Okay, and then the last clade, which I'll also divide up a little bit more out of the Mandibulata. Pancrustacea. It used to be thought that insects and centipedes and millipedes were close relatives. Now with the advent of DNA sequencing, being able to compare genomes, it's now understood that insects probably originated from this group called Crustacea. And so that insight has now rendered what was once a clade, or at least a taxon, that was a class in the Arthropoda, that was called Crustacea, is now paraphyletic. Therefore, including the insects, we can make it monophyletic Pancrustacea, right? Now, one of the apomorphies that once the DNA evidence showed that insects came from some group of marine crustaceans, then they started looking closer at the anatomy of these things, especially the structure of the brain. And crustaceans and insects have similar brain structures, not shared with other arthropods, but also they have compound eyes, consisting, and that's not just only within, like horseshoe crabs, I think they also have compound eyes, and even some trilobites had compound eyes, but they did not consist of structures called ommatidia. They were more like several simple eyes that are close together. So you had several separate individual simple eyes that still did not focus and did not form an image, but still being clustered together, those compound eyes would still have a little bit better discrimination in terms of being able to detect motion and light and dark, areas of light and dark. With an insect eye, or a crustacean eye, compound eyes sometimes have many, many ommatidia. In fact, a dragonfly might have somewhere upwards of 1,000 ommatidia. So an ommatidium is a unit of the eye, has a crystalline cone that forms the lens, and those cone cells, there's 4 of them, that secrete the lens. The lens does not focus. Okay, what this ommatidium is it is a long structure, nerve cells down here, light sensory cells at the very bottom, and a lot of times pigment along the side, so that, and it's immovable, can't focus, but when we have a curved structure with all these ommatidia, the light that can reach the nerve cells and trigger a response in the brain has to be coming in right straight. Any light that's coming from the side will not be detected. It's only coming in straight that it is detected. And so when you have thousands of these that are arranged in a arc like this, then the brain is picking up information of the light that's coming straight above, straight from that direction, and it's put together in the brain as a mosaic, somewhat like the little pixels on your computer screen. And so they're seeing bits of light, and it's fine enough that they probably detect figures, okay? They can see an image. And they can differentiate between colors. So this is the type of structure that happens to be in crustaceans, pancrustaceans. This is the apomorph, right? Now crustacea, this is mostly marine, freshwater. There's a couple, or one group of crustaceans that happen to be terrestrial, and that happens to be roly-polies. Okay, they're called isopods, but you probably would recognize them as roly-polies. Those are crustaceans. They belong to a different group. This is a big group, and it includes crabs, lobsters, krill, barnacles, and many others. And I say here probably paraphyletic. Get rid of that. It is paraphyletic. There's enough evidence now that what makes it paraphyletic is the exclusion of the insects. Okay, because insects have arisen within the pancrustacean. In other words, there's one group of one subclade of crustaceans that appears to be the sister group to the insects, and we don't need to worry about what that group is. So I'm just going to keep them just as a paraphyletic group, because there's too many clades within this group to deal with, right? They have two pair of antennae. Most have two pair of antennae, crustaceans. Insects are different. So there's one pair here, long, and another pair very short. That's mostly in the lobster crab group. And the number of legs is variable. Their body is also, most crustaceans have cephalothorax and an abdomen. However, as I said, there's many different clades there, and some of them have head, thorax, and abdomen, okay? And of course, compound eyes. Those that happen to be marine, living in the ocean, or in the water, even in fresh water, they breathe by means of gills. And many of these groups have their exoskeleton reinforced with calcium deposits, making it very, very strong. That's very much true of crab lobster group. They have very hard exoskeletons. Now, one monophyletic group that comes out or belongs to the pancrustacea that we're still going to emphasize it is the insects. So insecta, it's also called Hexopoda. Tagma, and I'm putting it here as apomorphic for this group. There's three body regions: head, thorax, and abdomen. The head has the sensory apparatuses, the antennae, the eyes. It's also where the mouth and mouth parts happen to be, followed by a three segments to the thorax. The head is probably fused with 6 segments fused together, and it's hard to tell the segmentation there. The thorax has 3 segments, each with a pair of legs. So thorax is for locomotion. Yes?
Does Crustacea have any apomorphs?
No, because I don't give apomorphism for something that's paraphyletic.
Oh.
The individual clades within it have apomorphism, but we're not going to talk about isopods or the other clades within it. The only one I want to talk about is insecta.
And would that
But for pancrustacea, yes.
like Crustacea would branch out of pancrustacea?
No, what we have with pancrustacea is a bunch of individual clades. Might be like this. You might have various branches. Where this branch right here is the insects. Okay, this group here, as a whole, is monophyletic. And there's apomorphies down there, and I gave one. The four crystalline cone cells in the Ommatidia. And there's other things here, mostly features of the brain. Okay, there's along the nervous system. A lot of neural apomorphies that belong here to this whole group. And so this here might be crabs, lobsters, over here isopods, and there's a bunch of others. Krill shrimp belong to a completely different clade. So there's a bunch of clades there, branches, but the insecta has arisen right within.
Oh, so Crustacea isn't like its own separate clade.
Now, we're just calling crustaceans just like the bryophytes,
Oh, okay.
okay? It's paraphyletic.
Okay.
Okay, it's any pancrustacean. Crustacea is any pancrustacean that is not an insect,
Okay.
okay? And so that's how it's working here. Because we go into all the different branches of pancrustacea, it would be too much, because there's about 10 of them, okay? And I don't think you want to learn any more, right? You have enough, right?
Maybe, yeah.
We still have deuterostomia to do yet, okay? So that's not an apomorphy, just mentioning it as a feature. Now with the insecta, this is monophyletic, okay? This is monophyletic, and having 3 tagma, head, thorax, abdomen, 3 pairs of legs, all on the thorax. So there is a prothorax, a mesothorax, and a metathorax, okay? So right behind the head, that segment, prothorax, 1 pair of legs, mesothorax, the middle segment has a pair of legs, metathorax is the last, and it has a pair of legs. Okay, and so 3 pairs of legs. Now, another monophyletic group within the insecta is Pterygoda. They have wings. So we go here, we have silverfish. Down here, something called springtails. And there's one other group, we don't need to worry about it. Here. Is Pterygoda. In other words, silverfish, springtails, and 2 other basal groups, they came from ancestors that never had wings. Okay, so not all insects have wings, but here they have evolved wings.
So they lose them like after?
The loss of wings has happened over and over and over again. Okay, but still, we can tell. Okay, so Pterygoda, these have 2 pairs of wings. 2 pairs of wings, and the wings. It's controversial where the wings came from. They may have come from flaps, ridges on the dorsal part of the exoskeleton in those segments, but it's not real certain.
You notice you spelled it
Pterygoda. Oh no, I left out the Y, or put in, yeah, the spelling on the screen is correct. Yeah. PT. Yeah, Pterygoda. PTER refers to wing in Greek. Okay, Pterygoda. Wings. And usually they're a pair of wings, one on the mesothorax and another pair on the metathorax. Prothorax doesn't have wings. Prothorax doesn't have wings. Now, as I said, many insects within the Pterygoda have lost wings, but we know from the structure of the thorax that they had to have come from winged ancestors. Because one of the problems with wings is that the thorax, especially the mesothorax and metathorax, they had to be reinforced, okay, in order to handle the flight muscles. And that led to a modification of the wing. In all arthropods. Usually the exoskeleton is somewhat like this, every segment. There is a few what are called sclerites that are at the base of the leg. Okay, they're hard places for muscles to attach. This is where the leg attach. And they have a tergum on top and a sternum down here. Now this is called a nodum. It's another word that still means the same thing. But on the dorsal plate, that is called a tergum, dorsally, and then ventral is a sternum. And that's pretty typical of most arthropods for every little segment. And if it's a segment with legs, then we have these sclerites. And then what we have here is a lightly sclerotized or lightly chitinized, okay, flexible membrane, okay? So that's membranous right there. And so, and that's flexible. Now the way in which the flight muscles in the insects work, usually the wings are up here, and most of the time they don't have direct flight muscles, okay? So the muscles are going up and down, okay? Attached to the sternum, attached to the tergum, and when these muscles contract, it deforms the thorax. It deforms the thorax in such a way that it creates a lever that makes the wings go up. Now, in order for that to happen, these sclerites had to expand. In the mesothorax and the metathorax, there is no flexible membrane. Okay, these sclerites have here the nodum. This is where the leg happens to be, wing right here, but all those sclerites have expanded. Then sternum down here to reinforce that whole mesothorax and the metathorax so that it's a pretty rigid box without any flexible membrane. All right? And for the flight muscles, because the flight muscles are going up, down like this. And in some insects, those flight muscles are really huge. Okay? So, this reinforcement. So, we look at a flea. Do fleas have wings?
No.
Fleas don't have wings, okay? But they have all this reinforcement. Now, how do you explain that reinforcement? Okay, it's because they came from ancestry where those that did have wings. Now, we don't have this reinforcement in the prothorax, incidentally. Okay, it's not there. All right? And other groups like ants have wings? Oh, but they do. They're reproductives. They do have wings. It's just the majority of ants are workers. They're the social insects where almost everyone is female. Okay, males are very, very rare. They only produce males just to mate. Then they kill them. Okay, this is with bees, social insects. Okay, that live in big colonies. All right, it's not true with termites. Termites are a little bit different. All right, but yes.
Why is wings considered an apomorphy if, like, silverfish number came from something with wings? Like if another.
Well, it's not an apomorphy for the insecta. It's apomorphy for Pteranodon.
Oh, okay.
And springtails, you look up there where I have springtail silverfish, they're outside of Pteranodon.
Okay.
Okay, they're a different clade. Okay, but they don't have, they are ancestrally without wings. Okay, and so these other groups, ants do have wings. Okay, the reproductives, they have wings, just that the workers don't. And even the queen ant, a new queen ant, will have wings. But after she establishes a new colony, she'll break the wings off. Okay, you don't have. Termites, okay? Termites, they also have wings. The reproductives do, but the workers do not. All right? And then lice, fleas, they don't have wings, period, but they still have this thoracic reinforcement. And furthermore, they arise in the phylogeny well up in the Pteranodon, okay, because they're positioned. And so the loss of wings is happening in common. So look at that thorax, especially here, the mesothorax, metathorax, they have those big plates. Okay, now, I said the arthropods, old Arthropoda, that the numbers of species outnumbers all species of life. If we take the insecta by themselves, they outnumber all species of life, including other arthropods. So this is one of the most successful. The number of species and number of individuals is any expression of evolutionary success. This is one of the most evolutionarily successful group of organisms. And this will give you just some insects. Silverfish, okay, and beetle, ants, praying mantids, dragonflies, and the beautiful flies. I like these flies. Okay, that's a midge.
It's not a mosquito.
That's not a mosquito, no. No, that's not a mosquito. That's a non-biting midge. Wings, it's a Chiroptera, belong to the family's Chiroptera. Okay. All right. Now, there's an issue with their post-embryonic development that happens. We have a couple of terms that are only true of insects. Namely, what is post-embryonic? Okay, the embryo develops in an egg. Okay, but then after it hatches out of the egg, it has to go through certain developmental phases in order to become and reach the adult. And so there's three different categories of post-embryonic development in the insects. Ametabolous, which is like all the other arthropods, but we still don't use this term ametabolous for other arthropods simply because they don't have the other distinctions like we're going to put up here shortly. So here with ametabolous development is that after hatching out of the egg, and this is true of silverfish and springtails, is that they look like a miniature adult, okay, except for they're sexually immature, all right? And as they go from molt to molt, and they increase in size, they eventually will reach the adulthood, and when they're sexually mature and can reproduce, and they continue to molt after maturity. That is way different than the rest of the insects. Because once, with the Pterocoda, once they have molded into the adult, that's the last molt that they ever have. But this ametabolous group keeps right on molding all throughout their life. Okay, so, and this is what it is like in all the other arthropods as well. However, because of these other two patterns, we make a distinction of the insects, saying they have these post-developmental phases. So we have another group here that is hemimetabolous. This is like grasshoppers, cockroaches, and termites, and true bugs. They happen to follow this. And this is only in the Pterogoda, okay, winged insects. So when they hatch, they look like, sort of like miniature adults, but they don't have full-fledged wings. They're also sexually immature, but they will have a pair, many of them have a pair of wing pads. Okay, these are embryonic tissues that are sticking out. You can see them. They look like a pair of real short wings. And they will go through several molts, and their last molt will produce full-fledged wings for those groups that still retain the wings. Okay, for those groups that still retain the wings, they will develop both with a pair of wings, sexually mature, and that's the last molt. They never molt again. Okay, once they reach adulthood. And so the immatures are called nymphs. So we just have two stages here, a nymph and an adult. Nymphs become adults. And so aphids, true bugs, cockroaches, praying mantids, they all belong to this particular type of group. And the third pattern, And this group is monophyletic. Okay, I'm not going to add another taxon, however. I'm just going to mention that within the Pterygoda, there's a group called Hemimetabula. And these have hemimetabolous development. Usually what hatches out of the egg does not look like the adult at all. Okay?
Like similar to like butterfly larva and then.
That's right. Okay? So usually they don't resemble the adult at all. They will go through different molts, and they have a particular stage called a pupa. Okay? So other insects, hemimetabolous insects, do not have pupae. This is a unique stage just for the hemimetabolous insects. And it's here that, this is a feeding stage. Okay, this is what they build up fat and food reserves. And here, the pupa does not feed. Pupa does not feed, but what's happening in here is larval tissues are being completely broken down and replaced by stems coming from stem cells, the adult body. Okay? Wings, they're internal. Okay, so in the larva, they have wing pads, but they're not exposed on the outside. They are inside. Okay, that's different than the wing pads in the hemimetabolous group. They are external. These, the wing pads, are internal. And they will, during the pupal stage, they will evert outward. Okay, and then we have the adult. Now, one of the advantages, probably the evolutionary advantage of holometabolism, is that there's no competition for food between the adult and the offspring. They usually feed on totally different foods, most of the time. That's not always, but as a general statement, I can say that. So they have these like stages, egg to larva. These are not called nymphs. So the word nymph, larva, they have different meanings in the insects. Okay, so this is like mosquitoes, beetles, moths, butterflies, bees, ants. They all belong to this group. Okay. And just the beetles alone, Coleoptera, that particular clade, beetles, has over 500,000 species worldwide. It's one of the biggest groups of insects. Okay, so that's all the phylogeny stuff for this. Just now talk about a little bit of the impact that insects have on humans. There are many blood-sucking insects, and therefore they are vectors of various types of blood-borne diseases. For example, malaria, that's spread by the bite of a female mosquito belonging to the genus Anopheles. And sleeping sickness, tsetse fly, that's another dipteran, and bubonic plague. This was a big deal in Middle Ages, started even in China, East Asia, all the way through Europe. Sometimes when bubonic plague came through, it was very fatal. Okay. Today, now what causes the disease is a spirochete bacteria. Okay, and it's spread by the bite of a flea. Now, the fleas are affected too, because what happens, the fleas, they're sucking blood, and they have all these teeth that are inside their throat, basically, and this bacteria turns into sort of a gelatinous mat. And so when the insect is trying to take a blood meal, it's hungry because that gelatinous mat of bacteria prevents the food from getting into its stomach. Okay? Therefore, it's very hungry, and therefore it'll jump off of the rats because it's not getting food from the rats, go to a human, and when it tries to suck in the blood comes up and hits that mat of bacteria, and they regurgitate it back into the human's body along with the bacteria. So this is how the bacteria gets spread. So it's still endemic in some places, like India. There's always cases of bubonic plague, and even in the United States, mostly in the Southwest, Arizona, New Mexico. Usually it's coming from flea bites, from fleas that happen to be on ground squirrels, chipmunks, and so forth. So when you go to the desert regions and try to feed those chipmunks and ground squirrels, don't do it, okay? Because you're making them be too friendly. And and they'll spread the disease. Filarial worms, okay, that's due to microfilariae for elephantiasis. That is due to the bite of an insect, a mosquito. Yellow fever and dengue fever, these are viruses, okay? So those are caused by viruses, once again, from the bite of a mosquito. In the case of yellow fever, it's a mosquito called Aedes aegypti, okay? So, and then many, many diseases, viral diseases of our crops are spread by insects. All right, furthermore, the insects, many of them, just only a little few of them will feed on our crops and cause millions of dollars of damage every year. And, but some are good. They're predators. They eat other insects. And so we sort of like them, including some of these pests. Okay, in fact, this brought about UC Berkeley. Used to be real big with what's called biological control. But in recent years, a lot of the entomology department of UC Berkeley has been dismantled, and now Davis is the main place here in California, Davis and Riverside, for entomology. But still at UC Berkeley, biological control of insect pests was highly developed there. And what is biological control? Where are these pests, why are they pests anyway? Usually it's because they are they're not endemic to this region. And so due to the movement of humans, we have brought them from their native habitat, where there was a lot of diseases and predators that kept them under control. And now we put them in a new space where they have no controls, and they go wild, okay? And they're spreading through our crops. So the whole idea of biological control is, first of all, to find out where did this pest originate? Where is it endemic? And to go there and try to study its biology to find natural enemies. And then see if those natural enemies are compatible to bring them here and release them. And so there's hits and there's misses, but this is still being used. One of the best stories here where this worked was back in the 1880s here in California with Cottony Cushion scale. Okay, now scale insects, they're Hemipetatus insects, but they will get on citrus primarily, oranges, lemons, and so forth, tangerines, and they suck the juices out of the tree, which eventually kills the tree. So this started attacking crops, citrus crops in Southern California back in the 1880s, and the entomologist here, forgot his name, oh gosh. Anyway, they sent their team to various places in the world trying to find out where this thing came from, because it's not native to North America. And they found it growing in Australia. But in Australia, it doesn't cause any problem. And they wanted to find out why. Is there a disease that keeps the scales intact? What they found is a Dahlia beetle, very much like the ladybug beetle. And they brought this here to California. It's originally from Australia. Mass reared it and released it. And it controlled the Cottony Cushion scale. It didn't eliminate it. It didn't eliminate it. The whole idea of biological control is not to eliminate, because if you eliminate the scale, these guys have nothing to eat, because these only eat that scale. And therefore all these will die, and you have no more control. And so sacrifice a little bit of the citrus crop, keep these guys around, low numbers, that will keep those around, and you keep it under control, okay? And that was the whole idea of biological control. And that was a successful story, because we still have Cottony Cushion scale here in California, and we have the Dahlia beetle as well. There is a group of wasps, so this is the Cottony Cushion scale, and the Dahlia beetle right there. This is the scale, and that's the beetle. Parasitoid wasps. This is in the group called Hymenoptera, that includes ants and bees and wasps. Okay, now these parasitoid wasps, what they do, and there's a big group of them, a lot of them are real small, is that they lay their eggs inside the bodies of other insects, okay? Including caterpillars. A lot of our crops are attacked by caterpillars, moth caterpillars, not butterfly caterpillars. But these parasitoid wasps usually have a long egg-laying device, which they stick like a hypodermic needle through the skin of the moth, and they lay eggs inside, and then go away. When the eggs hatch, the larva of the wasp chews on the insides of the caterpillar, eventually killing it, okay? And so, how many of you ever seen that movie Aliens? Okay, see, Hollywood, thought that was great imagination. These guys have been doing it for thousands of years, okay, millions of years, okay? And this is an ichneumoniid wasp. That's an aphid, and this is laying an egg right into it. Just comes up to it and jabs it, okay? And that's a caterpillar. That's laying an egg inside of the caterpillar. Okay, and then lastly, we also get some food from these things, okay? Products, honey. Okay, who doesn't like honey? Okay, that's nice. Comes from bees. Silk is a product that's produced by many insects, and you can eat the insects. Okay, here in the West, we sort of have cultural stigmas against eating insects, but they're high in protein. It's a good food source. And pollination. It's mostly this group of animals that are the main pollinators of angiosperms. And that's it for that clade. Any questions? Any slide you need to see?
Can I see 16?
Slide what?
16.
16?
60.
60.
Yeah.
Yes?
Can you explain a little bit more about the scleroids and how it impacts, like, how it has wings?
Oh, yeah. Okay. Oops. Most arthropods will just have terga, sternum, and a few plates around the base of the leg. The rest of this is flexible membrane. This is flexible membrane, okay? In order to operate the wings, you have to have these flight muscles to deform a box-like structure so that it can move the wings. This is not a box-like structure, because all this is like an accordion, okay? If you contract those muscles, it's just going to flatten it out, okay? It's not going to move any wings. So in the evolution of flight, these scleroids had to become big, forming solid plates, so that this whole thorax segment is reinforced into a solid but bendable box, but not real flexible, not like a membrane. So to move the wings, those flight muscles are pulling down on the tergum and causing it to be deformed, but it's not like a accordion, you're not completely flattening it out. And so in order for the wings to operate, we have to have that expansion of those scleroids to completely make that thorax region a solid box. Okay, now we can go to our last clade, then we're into physiology. The deuterostomes. I've already introduced deuterostomes. Okay, so this is our last group. I'm going to go from here. Everyone have this because I'm going to erase. Okay, so come over here and get a picture of it. Because I'll tell you what. I'll just move it over.
You just want to put it on the other side?
Yeah, I'm going to put it over here. Deuterostome here. Okay, so with the durostomes, already introduced them before. There's two major groups. One called Echinodermata. I already earlier gave the apomorphies for this. They're all embryological. Mentioned that they have early radial cleavage. Blastophore becomes anus. Schizocoelus coelom formation, and so forth. Okay, so with the deuterostomia, you have Echinodermata and chordata. These are the two groups that we'll be looking at, mostly looking at chordates. Okay, so Echinodermata, these are starfish and similar types of mammals. Sea cucumbers, sea urchins, this is a sea urchin, brittle stars, starfish, sea cucumbers, they're all marine. They're all marine. But they have a deuterostome development. Apomorphic for them is that their adults have radial body symmetry. Isn't this bilateria? Yes, it is. So this is believed to be secondary, okay? Because the larvae, after they hatch out of the egg, the larvae are bilateral, okay? So furthermore, this is not just only simple radial symmetry, it's pentaradial symmetry. For example, in the brittle starfish, there are 5 planes of symmetry, okay? You can cut it like that, and so forth. There's 5 different sections that will give symmetry. Okay, so secondary condition. They have a complete digestive system. Okay, so on the oral side of the body, there's a mouth. On the aboral side is an anus. Okay, now what we saw with the radiata, which had radial symmetry, way off of our board over there now, is that they only had a gastrovascular cavity, one opening. They didn't have a complete digestive system. These have a complete digestive system. Furthermore, for movement and also for gas exchange, used in feeding, they have a complex system here called a water vascular system. Okay, now, these things don't move real fast. However, a lot of these starfish, they're predatory. And what they prey on is clamps, bivalves. All right, so bivalves don't move too fast, so starfish doesn't have to be super fast either. But with their water vascular system, they have something called tube feet. They're not really feet, but here you can see it. So here's the arm of one of the arms of the starfish, but down the middle of that arm, on the underside, there is, or actually not the underside, that's the oral side, that's the side where the mouth is they'll have hundreds of these little flexible things that are called tube feet. These tube feet are flexible with sucker on the end, okay? But how they're moved and powered is a little bit by muscles, but mostly by the water vascular system, okay? By water pressure. Water pressure moves in. And so what this water vascular system happens to have is that on the aboral side, okay, next to the anus, there's a sieve-like opening that's called the madrepori. So this, like a little screen, this is where water will enter into the water vascular system. Then there is a fleshy stone canal. Okay, this here is showing the madrepori, then stone canal, and right in the middle there is a calcified circular canal called the ring canal. Okay, now from the ring canal there is also a calcified structure that goes down each of the arms of the starfish. Okay, this is called the radial canal. And along the side of the radial canal are tubes that connect to the tube feet. And so the water is being brought into the madrepori, through the stone canal, through the ring canal, down the radial canal, and it creates pressure in the tube feet. Okay, now, so it can walk with these 2 feet. And furthermore, the way in which they eat a clam is they'll put their arms on each side of the shell, and they can, with hundreds of 2 feet, suction cups go onto it, they can pry it open a little bit. And they have 2 stomachs, okay? First one is called cardiac stomach, and it can evert it out its mouth. And so what they do is they will slightly open the clam's shell, evert their cardiac stomach into the mantle cavity where it is releasing digestive juices. Okay? Eventually, those digestive enzymes are eating up the visceral mass and also the adductor muscles, and it weakens, and therefore it can then open the clam completely and finish the job. Okay? So those 2 feet are on the oral side. Okay? These things are moving mouth down. Aboral side is up. Okay? And down the middle of it is a groove, and when you come back from spring break, you'll see some of these, because I have a bunch of them in the lab. And that's all I'm going to say about that. Okay? The major, furthermore, let me go back. Separate sexes, they're dioecious. What they have going down each of these arms is their gonads, and they really can't tell males and females from each other. Okay? Their gonads look pretty much the same. Until they produce sperm and eggs, then you can tell them apart. Okay? So they have a pair of gonads, testes, ovaries, in each of their arms, plus a large digestive gland that produce a lot of digestive juices for the cardiac pyloric stomachs. Now, this is the group we belong to, the Chordata. And I'm going to give a bunch of Amphiboretes for this group. Okay? One is that they have what is called a notochord. This is primitively made out of cartilage, that type of, it's flexible, and it's a rod that is solid rod that is found between the gut, the digestive system, and the dorsal nerve cord. Dorsal nerve cord. Notice up to now, all the nerve cords I've been mentioning are ventral. This is a dorsal nerve cord along the dorsal side. Okay? And furthermore, that dorsal nerve cord is hollow and has a fluid-filled space in the middle of it. It's not a solid cord as in all the other animals we've been looking at. Okay? So this is anthropomorphy as well. Okay? Now, so we have the nerve cord, and the notochord is between it and the gut, the digestive system. So this notochord sort of serves as a brace. Furthermore, they have a muscular postanal tail. Okay, now what does that mean? It has a tail that goes posterior to where the anus is. In all of our other worm animals, nematodes, insects, earthworms, the anus is right at that's the last thing at the end of the body. There's no tail. That's the last thing there, okay? But chordates have a tail, okay? And now your book will say this is unique to chordates. Pharyngeal slits. It is not unique, okay? Because there's one other group, I'll put it up here, that is not the main group that we're going to talk about, but in phylogeny, it's right here. These are called Hemichordata. They are worm-like, they have a worm-like body, but they have pharyngeal slits. They're phalanges. And primitively, what these pharyngeal slits were for for filter feeding. Okay, and in fact, some other lower groups of chordates, they are filter feeders, just like the hemichordata. So what this is that in a digestive system like this, you have a mouth, and right behind the mouth is a region that's called the pharynx. And then behind the pharynx might be a stomach, okay, so forth. So this is reinforced by braces, okay, sort of cartilaginous braces, but they're holes in the digestive system, slits, we call them pharyngeal slits, and then also on the surface of the body wall. So as they take in water that has bacteria, other organic material dissolved in it, that water will move out of the pharynx here through those slits, and there's mucous membranes there which will collect the food debris, okay, and then they will exit out the body. The food that's collected on that mucus will continue on down into the rest of the digestive system where it gets digested. All right, so that's a filter feeding device. Hemichordates have it too, and lower chordates have it as well. So these pharyngeal slits in later, bigger chordates, they serve not only for filter feeding, but also for gas exchange. It's from these that the gills happen to develop from in a fish. We have them as an embryo. We have them as an embryo, but they disappear. Some of the bone, some of the slits articulated, we'll see that they have given way to our jaws. Okay, so I'll talk about that a little bit later on. Right, so pharyngeal slits. Yeah, so they're both for filter feeding, for gas exchange. The gills in a fish are derived from these pharyngeal slits. Okay, furthermore, another apomorphic, also in the pharynx region. Okay, there is a gland there. It's either called an endostyle, or when it's gathered together with some other tissue, it's called thyroid. And it functions with iodine metabolism. And it produces hormones. We'll talk about some of those hormones a little bit later. Another apomorphy. Now this is not in your textbook, and this is not in your textbook. We seem to be animals that have been compared to the rest of the animals kingdom. We're sort of flipped upside down. Okay. All right. We have a dorsal nerve cord instead of a ventral nerve cord. Ventral nerve cords are widespread, but this dorsal ventral inversion is. I mentioned Hox genes earlier, that these things code for proteins that diffuse through the body tissues, and these are transcription factors. Transcription factors that will bind to certain genes, and they're gradient dependent. And that set up the axes as to what is going to be ventral and dorsal, right, left, and so forth, body axis. Two of these proteins, one that's called chordin and bone morphogenic protein, their gradients establish what is dorsal and ventral. Okay, dorsal and ventral. In all other animals, bone morphogenic protein is produced by cells on the dorsal side of the body. And so as that protein then diffuses through the body fluids ventrally, its concentration becomes less and less. And this influences the expression of ventral cells to become the ventral part of the body. Whereas chordin has just the opposite gradient. This is the way it is in hemichordates. This is also the way it is in other animals. This up here, PS, that's pharyngeal slits. That's where they are in the hemichordates. Okay, only the chordates, hemichordates have pharyngeal slits, but they're on the dorsal side. So what is this dorsal ventral inversion is when you look at a chordate, the pharyngeal slits are on the ventral side. Furthermore, chordin has its highest concentration on the dorsal side instead of the ventral side, where this protein gradient between bone morphogenic protein and chordin is completely flipped over. So we are flipped over, deuteroskells. Okay? I think that's probably all the further I can get. Yeah, that's all the further I can get. Okay, so this is where I will stop.
What is bone morphogenetic protein?
It's one of those, what's produced by a Hox gene. And so it is expressed in the early embryo. It's transcription factor. Okay?
And what is the Hox protein again?
These are genes that code for proteins that control symmetry in the body.
Oh,
Okay, they determine their expression because they're not expressed the same all throughout the body.
so does everything under Eumetazoa have it?
Well, yeah, like all animals have some sort of oxygen, especially Bilateria. And even